Hi, everyone. My name is Theo Roth, and I am a professor at Stanford University and an investigator at the Art Institute in Palo Alto, California. I'm excited to be here today at this LabRoots Cancer Research Virtual Conference to tell you guys about comprehensive genetic manipulation of human cellular therapies and some of the underlying genetic engineering technologies that we've developed over the years to further empower genetic research in primary human cells, and especially for clinical translation of cellular therapies. So a central question for what a lot of us do is, how do you take a cell that you can get your hands on, and primary human T cells are easily accessible for both patients and healthy donors, and put in some new genetic instructions to get a desired cell state? And that desired state and desired set of functional properties may be different from one application to another, but the underlying principle is the same: that how can we understand what genetic changes to make to get the cell that we want for a given indication, for a given patient, for a given therapy. And so the taking cues from natural systems, the new cellular functions and new cellular states have evolved, innumerable times through our evolutionary past through the acquisition of complex genetic changes over the course of evolutionary time, and then more rapidly within individual patient through complex genetic changes within somatic cells, leading to the evolution of the new transformed cellular state that we call cancers. And so the genetic changes that both germline and somatic evolution use to acquire new functions come in a variety of different types or categories, and they certainly don't limit themselves to any one type. That might be single base pair mutations, knocking out a gene, knocking down a gene, or overexpressing a gene, duplicating genes to get more of it, or even combining multiple pieces of different genes together to make brand new genes. And all of these mechanisms are used often all together to drive both germline and somatic evolutionary changes in the acquisition of new functions. And so a central goal of what we do in the lab is, can we build technologies that allow us to access the same evolutionary potential of germline and somatic evolution, but direct it towards building new functionality or new therapeutic properties into helpful cells, into primary immune cells that can go in and actually match toe-to-toe with transformed cancers. And so those overlying question: how do you get it, in our case, how do you get a human T cell to do what you want in order to try to clear and the large problem is always large solid tumors. And so how do you get a primary T cell to clear solid tumors using genetic software? And so I'll talk about a couple of different kind of technical categories that we've focused on to do this. The first is integrating new genetic sequences into the genome at defined sites so that you can take advantage of natural regulatory and natural genetic properties. The second is a series of work that we've done to try to increase the scale of genetic sequences that you can integrate into the genome, with the idea that the more sequence you can put in, the more powerful you will be your genetic program, and the more powerful the change you'll be able to put into your cell. The third is, how can we scale up the testing of new genetic sequences so that you can test, instead of making one prediction and hoping that it works, test hundreds or thousands of genetic predictions or new genetic sequences and empirically determine and let the cell tell you which one works best. And then finally, how can we increase the power of these genetic programs by potentially having more or multiplex or combinatorial or more tailored approaches to putting exactly the right genetic sequence into a cell for the desired properties. And so I'll start with integrating with genetic software. And this is work that's now over 10 years old, with the initial development of CRISPR systems and application into primary human T cells. Everything that I'll show you today is based on this simple, cheap, extremely scalable ability to introduce new biologic macromolecules into cells of interest, in our case primary human T cells, using electroporation. And essentially you put your cells in a capacitor, you zap them, and pores form in the cells due to the nature of that electrical field. Those pores rapidly close, but during the time they're open, any surrounding macromolecules, whether that's DNA, RNA, or protein, can diffuse into the cell, and now you have delivered large amounts of cargo into cells. It comes at the cost of the viability, and that certainly some cells don't repair their membranes, but it's an extremely efficient delivery way and extremely scalable. And so starting out with doing knockouts, we put a lot of effort into optimizing and developing gene knock-in screening, such that we could integrate large new sequences into the genome of primary cells, specifically at the site that we want. And the way this works is that you use a CRISPR-Cas9 system, either an RNP or mRNA delivered, and that targets a double-stranded break for a single site in the genome. Now that single site and that double-stranded break needs to be repaired to maintain genomic integrity. And so either it can do the fast and dirty way of non-homologous end joining and smash the two double-stranded ends together. This often introduces mutations, which is how we are able to do gene knockouts. Or it can be integrated by homology-directed repair when we essentially trick the cell, and that into doing the the analogous slow but precise repair mode of homology-directed repair, which is essentially the cell thinking that it's going to repair its double-stranded break off of a recently copied, recently duplicated other arm of its chromosome, and so which during cell division is sitting right next to the template copy. And so when you have a double-stranded break that occurs during cell division, which is a common time for double-stranded breaks to happen, the cell can template off of the other chromosome to repair that seamlessly without mutations through homology-directed repair. We trick the cell by putting in large amounts of new double-stranded or of new DNA that we call a homology-directed repair template. And this new DNA, essentially, all you have to do is have the new sequence that you want to integrate flanked by what we call homology arms, which are the exact same sequence as either side of the double-stranded break. And so the cell template, the two arms of the double-stranded break bind by complementary base pairing to the two homology arms, thinking that it's the other chromosome, but then the cell seamlessly integrates the new DNA sequence in its genome. And so here's an example of integrating a GFP at a housekeeping gene called RAB11A, and at a high efficiency. So you can do this, rapidly across the genome. All you have to do is use a different guide RNA and then use different homology arms around your sequence. But here's showing live cell GFP fusion tagging of a whole bunch of different hallmark cytoskeletal or housekeeping elements that then these are confocal images of live primary human T cells that now have GFP fused to a bunch of different nice looking but hallmark cytoskeletal genes. But just to show you can go rapidly across the genome and integrate new sequence, wherever you want. And one of the powers of this kind of simpler mechanical delivery approach is that it's highly applicable across cell types. And so here's using more or less the same gene editing approach, same templates, guides, but different culture conditions, obviously, for each different cell type. But going across the hematopoietic tree, we can integrate new DNA at high efficiency across lymphatic cell types, across variety of T cell subtypes, including rare types like gamma deltas or regulatory T cells. You can do it in HSCs, but also even including iPS cells as well. So the being able to integrate new DNA at specific sites gives you a whole bunch of potential. And just to highlight a recent story that we and Justin Equim's lab at UCSF had a similar story this year around showing how you can actually create advantages in how you select for cells just by choosing a different site to integrate. And in this case, normally we integrate new DNA into existing exonic regions, but by moving the site you integrate into an intronic region, you get this nice property in that all of those non-homologous end joining mutations that are essentially like failed HDR outcomes are sitting inside an intron, which gets spliced out of the final transcript, thus allowing you to maintain expression of the endogenous gene when you don't have successful integration of your new template. But when you do have successful integration, now you can seamlessly and touchlessly negatively select those cells just by depleting on the cells, in this case, that still express the TRAC gene. And this, you don't have to include any additional sequence. Just by choosing where you integrate, you have this nice property that you can easily select and highly enrich your edited cell population without having to put any new sequence in. And so That's just a couple of vignettes around how you can integrate new DNA with endogenous software to get new advantages. But I'll quickly go over kind of the developments of the last 10 years and the continual increases in the size of gene knock-ins that we've been able to do in primary immune cells. And so 10 years ago, we were really excited to be able to put one base pair in. But these technologies have rapidly progressed, and over the course of the last 10 years, we've now reached up to over 10,000 base pairs being able to be integrated at a single site in the genome of primary human cells. And we and other groups have contributed a lot of different optimizations to how to do this. And so you can put a huge amount of DNA essentially wherever you want in the genome of primary human immune cells. That's not limited to immune cells either. We've been able to do this in iPSCs as well. And so the efficiency of integrations have dramatically increased. In this case, this is like a kind of like a preclinical demonstration series of 24 patients just to show the variability of this. And integrating a 5,000 base pair template into the track locus, we can get on average about 50% integration of this using current protocols. And then you can enrich this to even higher if you want again, without even touching the cells. And so we can put huge amounts of DNA into primary T cells now for pretty cheap. You just have to build the DNA sequence, and you press a button to electroporate it into the cells. And so our problem has shifted from how do we put more DNA in to how do we know what the right DNA sequence to put in. And so in that case, you can make individual predictions about what sequence will work best or worst. But we focused a lot in the last 5 years or so on developing a series of ever more powerful genetic integration or genetic screening tools where you can screen simultaneously many DNA sequences together and let the cell tell you which one does best for that given application. And so some of these earlier protocols have been published, and so I'll just breeze through. But essentially, we instead of introducing one DNA sequence into a set of human T cells, we introduce a library of initially dozens, then hundreds and thousands, and now tens of thousands of different sequences simultaneously, and each sequence is tagged with a unique barcode. And now, because we're doing a targeted integration at a single site in the genome, each cell will successfully edited cells will integrate at one or at most two, if it's biallelic integration, members of the library, even though many members of the library go into the cell with the electroporation. So they end up only expressing one, and the one they are expressed is associated with a barcode that is integrated into their genomic DNA. And we can either do simple amplicon sequencing, using a little trick of homology-directed repair to get selective amplification. And you encourage you to look through the paper for all those molecular details. But you can pair it with single cell readouts. So you can get high-dimensional single cell readouts of a whole cell transcriptome or accessibility profile or whatever other multimodal single cell workflow you're using that involves RNA capture. And you can associate all of those cells with the large DNA sequence that you integrated in. And so just to show kind of an example of how you can use this, this is as in vivo gene integration pooled knock-in screen with single cell readouts. And if we do, and so this is in a melanoma solid tumor xenograft model, but in this case we put in a small library of about 36 different genetic sequences that would potentially improve the function of a CAR T cell, or in this case a synthetic NY-ESO TCR T-cell against a NY-ESO1 positive target melanoma cell line. And on the left here is what you would get by traditional abundance-based screening. And so we find a variety of constructs that enrich in the genome or inside the tumor relative to the input population compared to controls or the rest of the library. But we can also pair these with single cell readouts, which gives you a much more powerful way to look at this. And this is looking at just the transcriptomes of the input ex vivo expanded cells and looks very different than the tumor infiltrating lymphocytes that were pulled out of the tumor. But what we can do is we can go into this TIL population, and because we have single cell profiles that are linked with every genetic perturbation, you can almost do kind of flow cytometric style gating on whatever population you think is important for your given application, and whether that's you want naive or stem-like cells, or proliferating cells, or cells that are activated, or cells that are expressing effector cytokines. You can go through however you want. You can define gene signatures of hundreds of genes if you like, and you can find which constructs or which genetic sequences enriched or gave you preferentially that phenotype. And this is, we have that for every construct, and this is just a subset of that, showing only the constructs that enriched in abundance in the tumor. And what we can see is that these constructs, a set of them that has a TGF-beta receptor chimeras, behave extremely differently than a TCF1 or TCF7 overexpression construct, even though both of these sets increased in abundance in the TILs. And so you can highlight extremely different functional profiles, which may be associated with very different phenotypic performance. And so we've continuously improve these methods. There's a lot of molecular biology under the hood here, but essentially, we've been able to scale this from dozens to then, in this story from 2 years ago, to hundreds, and then now up to thousands and thousands of gene sequences. So I encourage you to look up some of this previous methodologic details. But essentially, we've built multiple generations of these technologies that initially allowed us to integrate single new gene sequences at a desired site in the genome of primary human cells. We could then scale that up to doing essentially any easily culturable primary immune cell, and we can integrate larger sequences. So you can put 2 or 3 genes. Developed initial pooled knockin technologies that allow you to screen up to about 100 genes at a time, and then improved workflows that allowed us to go up to 10,000 genes at a time. But even with all of this genetic power, we're still left with this question, and you talked about it at the beginning, is that germline and somatic evolution don't limit themselves to just integrating and overexpressing like one new gene. They do genetic perturbations across all types, whether that's knockouts, knockdowns, overexpressions, domain fusions, on-coding elements. All of these happen. They happen at scale. Hundreds or thousands or millions of different genetic mutations are being tested simultaneously, essentially, in the competitive evolutionary environment of a developing tumor. And then they're happening in innumerable combinations as well. And so the last couple of years, we've kind of gone back to the drawing board and said, can we build a genetic screening system that allows us to have the same genetic screening potential as germline or somatic evolution? Essentially, can we screen complex genetic perturbations of any type, in any number, at any scale, in any combination, and do it all simultaneously and link that with abundance or more powerful single cell readouts? And so the method that we've now developed that should be out pretty soon is we call CRISPR-all. And essentially it takes this dramatic proliferation of gene editing tools that have been developed over the past 10 years through using CRISPR systems to do all of these different types of genetic perturbations. But these existing technologies, even though they're highly scalable, are very bespoke in that they allow you to do lots of one type of genetic perturbation, but they don't allow you to do multiple types, let alone do any type in any combination all together simultaneously. And so we've built this methodology called CRISPR-all that is kind of rests on the underlying assumption that, look, like all of these different types of perturbations ultimately are encoded in DNA, whether that's RNA effectors, whether that's new genetic new proteins that are going up, or it's guide RNAs, shRNAs, non-coding elements, all of it, or barcodes and all their different varieties. All of them are encoded by DNA. And what we've developed are methodologies that allow us to integrate very large sequences of DNA in large pools. And so we did a lot of work in kind of the genetic architecture of this, but essentially have built standardized modules that encode for each class of major class of genetic perturbation that can be arbitrarily combined together. And so what this allows you to do, and they all have a unified barcoding structure such that you can test any perturbation class against each other simultaneously and read barcodes out in the same way. But you can also do any combination of perturbations together at the same time, and any combination builds up a unique barcode that allows you to read out that set of perturbations. And so, again, a lot of molecular biology that goes in under the hood, but essentially these constructs can be iteratively and modularly built through successive rounds of restriction, digestion, and ligation cloning. It's all relatively straightforward stuff. But essentially, the functional parts build up on the left side, and then barcodes build up on the right side as you go through indefinitely repeatable rounds of cloning. And that allows you to build up complex arrays of genetic perturbations, all with a single unique barcode that tells you exactly what happened. So just to show you that we can do this, here's a bunch of examples. Here's 5 genes being upregulated in primary T cells. Here's 2 full-length genes, but then breaking up a couple of genes into individual domains that then get spliced back together to form the full GFP and mCherry in this case. Now here's 2 genes being overexpressed with 3 genes being knocked out. Again, all of this happens in one DNA sequence that gets integrated into the genome and has a unique barcode at the end. Here's 2 genes, but maybe you don't want to knock genes out, you want to knock them down. Here's 2 genes being overexpressed or knocked in with 3 genes being knocked down. And of course, the point is that all of this can be combined together. Here's multiple genes, multiple domains, and knockdowns and knockouts, all happening simultaneously in the same sequence. And so we think of this CRISPR-All system less as another CRISPR screening technology, but actually more of a genetic programming language or a genetic perturbation language analogous to computational programming languages. And that essentially it allows you to input a standardized high-level, plain English description of a cell and say, I want to knock in 2 new genes and knock out another one and knock down a 4th one. And what CRISPR-All does is converts that into a standardized syntax, and then you don't need to know the molecular biology rules underneath this because there's a standardized encoding of how all this works, and that builds up the series of functional elements that allows you to generate these perturbations. And so that encoding converts directly into a DNA sequence, which then if you integrate that sequence into your cell, and we do it by electroporation with a targeted knock-in, but you can do it by other methods as well, gives you a cell that has the desired changes that you want without you having to know all of the intermediate steps. In the same way that you might write code for a program in Python or R, or increasingly just typing in to a large language model what you want the code to do, and ultimately you get a program that does what you want. And you don't have to know about machine language or assembly language code or the binary sequence that's actually going to be loaded onto the CPU and how that's going to execute to give you the output. And so we think we kind of hopefully built a similar version of that for genetic perturbations, which allow much greater access to much more complex perturbations, the same way that computational programming languages allow you to access much greater complexity of programming tasks without having to know the underlying details. And so just to show kind of like the full generalizability of this, here is every possible or every valid combination within the CRISPR-All syntax of 4 different functional modules, along with 2 genes being overexpressed as the base. So all of these are 6 function modules with every combination of a variable of 4 sitting in here. That's like 4 genes going up, 4 genes being knocked out, 4 genes going down, 2 genes up with 2 genes knocked out, all these different combinations. And what we can see is like in the center column here is the predicted genetic program that we expect in the cell. And then on the right 2 are the observed program that we actually see, and this is based on protein expression in single cells by flow cytometry. We can in successfully edited cells that have the constant gene knocked in, we observe essentially the exact same pattern as predicted. And then in unedited cells, we don't see any of these perturbations. So it's extremely clean. So this CRISPR-All system allows you to have a unified genetic screening system to potentially build much more powerful genetic programs of combinatorial complexity arbitrarily across perturbation classes. And so just to show a couple applications of this into some immunotherapy work and how you can use this, one initial way you can do this is, well, now you can screen simultaneously in the same cell population arbitrarily across perturbation types. So you can directly compare in the same experiment, in the same donor cells at the same time with no batch effects, gene overexpression, by domain engineering, by gene knockdown, by gene knockout, all together simultaneously at scale. And so as an example of this, we've built a meta library of genetic perturbations that have been proposed or tested clinically for TCR and CAR T cell therapies. And there's a huge wealth of literature here, and we've found over 500 studies before our cutoff date, about over 75 large-scale primary mouse and human T-cell screens, a whole bunch of different CAR engineering strategies that have been used in clinical trials. And we built a single meta library across all of these different perturbation classes so that we could test all of, like, essentially the entire wealth of what's been discovered in this T-cell engineering field. We can consolidate it into a single library that you can do screens on in your assay systems. And so in this case, this is being run with in a chronic stimulation assay against live NALM6 leukemia target cells at a stress dose of 1 to 8 E to Ts, with the T cells expressing a CD1928 zeta CAR unless they have a full-length CAR variation. And what we can see is that we can find a variety of hits that come out, but the hits come from a whole bunch of different perturbation classes. And then we can actually directly compare the magnitude of effects and the differential types of effects from all of these different perturbation classes together. And just here's now all the different perturbation classes broken down. But again, these are all run simultaneously head to head, so the data is comparable. And we ran this, we've drastically lowered the cost and made it more simple and accessible to run these. So we've run the screen in 8 donors, and we can do it in many other screening settings. And so this isn't to say that, like, the construct that does best in this one screen is the thing that is the best construct for every CAR T cell therapy possible. But there's going to be differences. You run a different assay, you're going to get different results. But essentially, this allows you to start going systematically across any functional property, any environmental setting that you think is relevant for your clinical indication, and screen simultaneously every proposed genetic perturbation to improve T cell function that exists in the literature. And you can do all of it simultaneously to rapidly prioritize the most powerful functional changes possible and move those forward clinically, as opposed to testing narrowly within one perturbation type, which is the traditional methodology. And so last minute or two, just to highlight a couple extensions of this. All of this is compatible with single cell sequencing. So you can take pools arbitrarily across perturbation types, and you can link the perturbation with single cell sequencing simultaneously for gene knockouts, gene knockdowns, gene overexpressions, domain engineering, whatever you want. You can do it all simultaneously, and you can do it in any combinations of these as well, and get single cell readouts from all of these. And go to town with however you want to analyze that data to prioritize things. The last thing I'll show is just that that was showing just all individual perturbations being compared head to head. You can also screen all of this stuff combinatorially against each other. And so you can run complex perturbation screens. In this case, this is CAR signaling domains with gene overexpressions, with gene knockouts, with gene knockdowns, and over 10,000 four function combinations of all of these. So each construct has a different signaling domain, a different gene being overexpressed, a different gene being knocked out, and a different gene being knocked out. And you can screen all of this simultaneously together, and we get all kinds of interesting patterns and much more powerful things in this assay with a proliferation readout. Again, not saying that that's necessarily the end all be all of that. These are the things that should go forward clinically in every indication, but we can find extremely consistent and powerful combinations across perturbation classes that have much more powerful genetic or functional changes in these 5 donors that we screened than individual perturbations do. And so, I'll leave you with that. This is our CRISPR-all methodology. It's kind of like our summative of technical approach to primary human immune cell genetic perturbations. Allows you to simultaneously screen across perturbation classes and do so combinatorially in large complex networks. And so, kind of touched on a couple of themes about how you can build complex and hopefully productive and ultimately curative cellular genetic medicines by integrating new genetic sequences with endogenous software, by increasing the scale and ultimately the power of the genetic sequences, and using high throughput screening approaches to rapidly screen genetic sequences together simultaneously. And so with that, have a great lab that we just got started recently at Stanford. But huge thanks to our variety of funders who have been extremely generous in funding this kind of ambitious technology development work, and the great group of folks that we have at Stanford, and a variety of collaborators both at Stanford, ARC, UCSF, my former home, and throughout the country. And so thank you, and hope you guys enjoy the rest of the conference.